Hello everybody, and welcome to my presentation. I am Matti, I'm from Finland, and right now I'm a PhD student at the University of Graz in Austria. And today I'm going to present some of my research about how ants are affected by pesticides. I think we found some pretty interesting stuff, so let's get to it. And first, a reminder about ants and ant colonies. Usually in any ant colony, there are female workers, then there's the queen or multiple queens. Usually there are no male ants present in the colony, except for the time when the colony is producing the new sexual members of the colony, which are the new queens and the male drones. The queens and the drones usually have wings, and they leave the colony to find a new one. But sometimes the queens may also stay in their home colony. In Cardiacondyla obscura, which is the ant that we are studying, and also what you see in these pictures, a colony may have multiple queens. So sometimes she will stay in her home colony. In these ants, also the new males may sometimes stay in the colony, which is not very usual. And it's pretty interesting. And I have actually a poster about some of the male stuff. And check that out. It's pretty interesting. But that's not the topic for today. So let's continue talking about pesticides. In today's agriculture, pesticides are widely used, and they, of course, help making the yields bigger, but they also may have effects on organisms that are not the intended targets. It's hard to say exactly how much different animals are exposed to pesticides, but residues of pesticides can be found on the plants and in plant nectar and in the soil. So it's safe to say that animals like ants that are living in the soil and running around on the plants are exposed to the pesticides. But not too much is known about how ants are affected by sublethal doses. So we exposed our ants to two common pesticides to find out. And the pesticides that we used are thiacloprid, which is a neonicotinoid insecticide. It is used, for example, against aphids, so it is harmful to insects on purpose. There is some evidence that sublethal doses may affect learning in ants or in insects, for example. And the other pesticide is glyphosate. It's an herbicide and it's not supposed to be harmful to insects, but it may still well have some effects because it has been shown to be harmful to some microbes. So we set up experimental colonies, and this is how one colony would look like. It's a 9 centimeter petri dish with a plaster covering the other half, and there would be 20 ants and 1 queen living in one of these colonies. We have a little piece of tin foil there to cover this little nest cavity that we gave the ants like as a shelter. And food was given on these tiny metal discs. We gave them honey and a protein food item, which is a cockroach leg in this case. And we changed these food plates 3 times a week. To provide water, we kept the piece of Wettex moist. And the pesticide was mixed in the honey, so the ants were exposed to the pesticides in their food. And these ants are really tiny. So there, the big arrow is pointing at one. I'm not sure if you can see it, but these are really tiny ants. So we had a bunch of these colonies in 4 different treatments. The control colonies, they just got normal honey. The glyphosate colonies got honey with glyphosate mixed in, and thiacloprid colonies with thiacloprid mixed in. And then we also had this combination treatment with both of the pesticides mixed in the honey. And we used doses that are maybe a little bit high, but still something that you might find in the environment where these pesticides are being used. And we had these colonies running for 12 weeks. And after 12 weeks of exposure, I measured the size of new workers and new queens that were born in these colonies. And we also measured the relative number of two endosymbionts called Wolbachia and Westerberhardia. And we used these two just because we just happen to know that they are found in these ants. They are not necessarily harmful, might even be beneficial, or just doing their own thing. I think you maybe have heard of Wolbachia because it seems to be everywhere. Okay, but now I want to show you the method that I used. And first, here's I want to show you how I did the size measurements. So I used this really fancy microscope to measure three things in the ants. I measured first the width of the head, just above the eyes. Second, I measured the widest part of the thorax, which is kind of the back of the ant. And finally, petiole, which is the small, round, very mobile part of the ant abdomen. And to measure the endosymbiont numbers, we used qPCR and sampled dark worker pupae from the colonies. And it is the last developmental stage before the ants emerge as adults. The endosymbiont amounts were normalized to the ant reference housekeeping gene elongation factor 1 alpha 1, if that happens to say anything to anybody. And also a word about statistics. So I used linear models to analyze the numbers. The two pesticides were the fixed effects, and I compared also models with and without interaction. But because there was never significant interaction, I left it out. And in the size measurements, I had sometimes multiple samples from the same colonies. So I used a mixed linear model with the colony as a random effect to account for pseudo-replication. Okay, we are ready to see our results. And let's look at the endosymbionts first and start with Wolbachia. So in this graph, we see a measurement of the endosymbionts in the 4 different treatments. And the higher the box is, the higher the number of endosymbionts. So the lowest boxes seem to be glyphosate and the combination one. And when we look at Westerberhardia, it looks very similar. So what this means then is that in the colonies that were exposed to glyphosate, meaning the glyphosate treatment and the combination treatment, the endosymbiont numbers seem to be lower. And that's also what the model says. Glyphosate is indeed the only significant effect here. So less endosymbionts in the glyphosate treatments. Okay, so let's go to the size measurements and the worker size. Here I'm showing the head size measurement first. And again, you have the 4 different treatments, and then you have the head size in micrometers. I think you can already see what kind of trend we might be having here. So it seems that the new worker ants are smaller in the head in the colonies that were exposed to thiacloprid. And when we look at the thorax and the petiole, we see that the graphs look kind of similar. And that's also what we found in the models. Thiacloprid is a significant effect in all of these. And I think it's safe to say that ants in the thiacloprid treatments are smaller worker ants. But the queens, in the queens, the story looks very different. If we can look at the head size first, I think you can clearly see that there is the pesticides are not definitely making the queens smaller. There's no significant effect here on the head size, and also not in the petiole size. But when we look at the thorax, it seems that the thorax is actually bigger in the treatments that contained glyphosate. And that is indeed a significant effect. So we can say that in the treatments containing glyphosate, the thorax of the new queens was bigger. Okay, so what did we learn here? I'm just gonna sum up the results that are, in my opinion, are the most important. So first, the endosymbionts were reduced in the treatments that were containing glyphosate. Second, in the thiacloprid treatment, the worker ants were smaller, but there was no effect on the queens of thiacloprid. And third, in contrast to the second part, is that in the glyphosate treatment, the queens had a bigger thorax, but glyphosate didn't have an effect on workers. And why might this be then? So now it's, of course, time to enter the magical kingdom of wild speculation. So as I said in the introduction, glyphosate is known to not only kill weeds, but it is also harmful to some microbes. It has been shown, for example, that it can mess up the microbiome of the honeybee gut. So it's not maybe that surprising that it might also be harmful to some ant endosymbionts. And why would the worker ants be smaller? I think this is just a resource allocation thing. The ants need to get this harmful substance out of their system, and that, of course, takes up resources that could be otherwise used for growth. But it's very interesting that the queens were not smaller. And in fact, in the glyphosate treatment, the thorax of the queens was bigger. I think this is maybe the most interesting result here. And it's also maybe requires a bit more speculation on why I think that might be the case. So let's get to it. So first of all, the thorax is the part where the wing muscles are in the queens. Now, I think that the pesticides are creating a bad environment for these ants to live in. And if the queens have bigger wing muscles, it would make it easier for them to fly away from this awful place. So it may be that because the ants are living under bad conditions, it would make sense to leave that place, and therefore there's investment in the wing muscles. And the thorax would then be bigger, of course, because there's bigger wing muscles. And even if it's just speculation at this point, this is for me the most interesting thing in these results, and I think something that I would really like to look more into. So this is what I will leave you as a speculatory take-home message of my presentation. Perhaps exposure to glyphosate makes the ant queens invest in their ability to leave the bad conditions, and thus build bigger wing or flight muscles, and that is why the thorax is bigger. Okay, that's everything that I have for you today. I'd like to thank you for listening, and also the people involved, and of course my funders. And now I think I still have a couple of minutes, so I will be very pleased to answer any questions that you might have. Thank you.